much. Sorry for this inconvenience. Okay, so just let me tell you a little bit in the next 20 minutes what we are doing actually in the NERV team. Actually, my name is Fabrizio De Vico Fallani. I'm head of research at INRIA. I'm leading this relatively new team that has been created a few years ago, whose focus is on systems neuroengineering to model and interface brain networks. So perhaps first things to say is that we are an INRIA team, but we are actually physically located within the Pitié-Salpêtrière Hospital in Paris, which is a quite big hospital, and inside we have the Paris Brain Institute. So that is an institute hosting several research teams working on neuroscience from really different aspects, from basic neuroscience, like really focusing on genetics, molecular biology, up to clinical and cognitive neuroscience. So this is a great opportunity for a methodological INRIA team, which is mostly applied mathematics, to translate and any theoretical modeling and results into the real world. Indeed, the team is quite multidisciplinary. This is a nice picture we took last year. And actually, there are many disciplines that that constitute this team. And very briefly, the team is composed by 5 permanent researchers, 2 from INRIA, 2 from CNRS, and then we have an associate professor that just joined our team recently. There are 20 non-permanent members. We have postdocs, PhD students, and as I told you, we are really quite multidisciplinary profiles from mathematics, physics, and to really psychology and neuroscience. And you will see this multidisciplinary profile and expertise actually needed for achieve the goal of our team. Now, very broadly speaking, the team is interested in 2 types of research goal. The first is more on system neuroscience, and the second one is on the development of brain-computer interfaces. Now, I'm going to give you a little more details about these 2 topics, and you will see that in the end, even if these 2 research topics look separated, they are indeed very quite intertwined. And of course, system neuroscience is much more theoretical, while brain-computer interfaces is much more experimental. So we have a peculiar characteristic as an INRIA team that we also do a real experiment on, in this case, on human beings. Now, the first big questions we have about system neuroscience is how can we model the brain structure and the brain dynamics from a network perspective. Indeed, we know that the way the brain is structured, anatomically speaking, but also the way the brain is functioning in terms of functionalities, it can be really seen as a network. And here I'm just representing it on a cortex model of a real human subject. This model is obtained from MRI images, and here you can see you have different points or different colors, and these points, these nodes, represent different regions of the brain. Now you see that the connections, they are connected in a very specific way. So the connectivity between those nodes is not a trivial connectivity. So there is a lot of heterogeneity in the way these connections are distributed in the brain. And our goal is really to try to understand how the brain organizes and reorganizes in brain diseases in terms of this network. To do so, we actually need to make a move from and translate this practical problem into a theoretical one. And the way we do it is actually making an abstraction and transform and thinking and see this network, this brain network, as a graph, which is a mathematical object. I'm sure most of you know. And then we are interested in understanding how we think about this network, how can we identify which are the most connected nodes, how efficiently they exchange information, which is the tendency of the nodes to form specific model, and how all these, let's say, features of the networks correlate with the behavior of a person or with the presence of a specific disease, or how can we use this information to predict how a specific disease will evolve, and all these kind of questions. So the methodological framework that we have set in the last decades in the team, it's here depicted in a very simple format, but I hope digestible for those who are not familiar with this kind of approaches. And the pipeline is as follows. So we start with data that are experimentally acquired in vivo, typically from human beings, but sometimes we are also interested in gathering data from other animal species. But here you can see that the typical setting for a human being, where you have neuroimaging data. So the data can be signals or can be pictures, and are recorded through different sensors that can be on the scalp, for example. In this case, we are talking about electroencephalography, or we can use more sophisticated machines, big machines like magnetic resonance imaging, to access how the brain signals change over time inside the brain. What we are interested in, the first thing, is that actually the network, the brain is not, to get a brain network, you actually have to infer it from those experimentally acquired data. And a lot of expertise of the team is how to transform these signals, this time series, or those images into a network, into a graph. To do so, we have implemented a certain number of methods. Here I'm just showing a few of them, but the challenge in this point is really how can you handle and how can you derive reliable networks, knowing that the data that you acquire are highly complex and the signal-to-noise ratio is typically very low. Once you get the networks, then we have the questions: how can we extract information from these networks, which can be very complex, lot of nodes, lot of connections. Of course, visually it is impossible to understand what is going on. So we need to transform and take this network that here is identified by the letter A, the density matrix A, and apply some functions in order to transform these complex object in a few numbers that quantify a specific property of the systems, like, for example, who is the best connected node, and other properties like this. So we generally call it a graph analysis and modeling part. We also introduce a number of methods to try to model the way that is the best possible this kind of object. And of course, once you have extracted these metrics, you can inject and you can use these numbers and input them in a statistical framework, so that you can understand how those network properties differ between different cognitive conditions, or between different type of populations, like, for example, healthy or diseased people. Now, what is important in all this pipeline is that this network approach really allows us to have to give an interpretation to any possible difference we may find between a healthy and diseased population. So just to make a parallel with a sort of black box approach, where you just input the data, you let your artificial intelligence algorithms to extract some properties, make the discriminations between two different kind of populations. But then the problem from a neuroscientist and from a biology, The first things they will say is that what is the mechanism that allows us to make this discrimination? Now, this network approach allows you to be very specific in terms of those network mechanisms that discriminate between different populations. Say, for example, you can identify that what makes the difference in a specific disease as compared to a healthy population is the fact that there is one specific node, one specific region of the brain, of the brain that significantly changed the number of connections with all the other regions. And of course, you might have much more sophisticated mechanisms, but this number of connections is perhaps is the easiest one, but is the most intuitive to understand. Now, of course, here the situation when we talk about neuroscience and the brain gets complicated, because here I'm making a lot of assumption in the picture I did before, like, for example, that the graph is static, the network does not change over time, and of course we all know that this is not true. The brain network exhibits significant changes and fluctuation over time. That is to say, the way the different brain regions exchange information and get connected is not the same, and changes across different multiple temporal scales. And you can have a similar reasoning in terms of spatial scaling. So depending on which is the spatial scale you're interested in, the network is different. If you are looking at connectivity between neurons or between ensemble of neurons, the network can change. And of course, to take into account all these multi-scale organization of the brain, we need to develop more sophisticated tools based on complex system science. And this is actually another research line in the team. We develop methods to really understand the network dynamics in terms of temporal evolution. And we applied this, for example, in a recent paper to understand how the brain, the nervous systems develop from the early days until the adulthood. We can also, we have introduced methods to not only to characterize the networks, to extract features, but also to be able to control the dynamics taking place over the network. So it's a sort of taking results from control theory and adapt those results from control theory in the context of networks. On the other hand, we have also introduced, and we are really interested in looking at how the network structure can be modeled according to different layers of information. In particular, in the brain, we know that two same areas of the brain can exhibit different types of connectivity. For example, if you think about the way they are anatomically connected and the way they are functionally connected. So the former is more the highway that is present in the brain, and the second layer will be more the traffic that is present over the layer. And of course, having not having a connections in the highway does not imply that you cannot have a connections in the functional layer. So you can see how the situation gets complicated very easily. And we also develop methods to better integrate information from networks who exhibit different layers or levels of connectivity. We have also developed a way to try to simplify and apply linear algebra methods in order to better understand how can we embed these network spaces in a low-dimensional space where you can more easily treat and manipulate those networks. And of course, we are also very much interested in trying to see how can we integrate this network information from a machine learning perspective, and also integrating different types of modality, not only how different regions of the brain are connected together, but also how the brain and other organs can actually interact and how those interactions, like, for example, between the brain and the heart, can give rise to a better description, a better modeling of a specific pathology or a specific disease. So that was the first part, really, I have to say, really theoretical. I didn't have the time to, of course, I don't have the time to go into the details. And here we have the second research line of the team, which is more experimental, and is devoted to the development of non-invasive brain-computer interfaces. So what is a brain-computer interface? Is actually a system that allows you to interact with the external world by only using brain activity. And the way you do it is actually creating a sort of bypass between the brain and any external devices. So instead of interacting by using the typical skeletal muscle system, you actually don't need to move any muscles. You just think about making some movement, for example, and the system would be able to understand and, let's say, detect what are the brain changes associated to this mental intention and translate this into an external command. And here I'm going to show you a video of a recent experiment we did in the lab. You can see the subject is wearing a cap. There are some electrodes on the scalp. Everything is non-invasive. The electrodes, those set of electrodes is called electroencephalography, and those sensors, what record is the electrical activity produced by the neurons in the head. So we collect on real time this brain activity, those brain signals. Those signals are sent to a computer, and in the computer we have a certain number of more or less sophisticated algorithms that detect in real time what are the changes in those brain signals, and how can we determine if those changes are associated to the true mental intention of the person. If the classification is correct, then we deliver a stimulation to a robotic arm, and the goal of the subject is to grasp a can on the table. So here you can see how it works.
Yes, it's working, but I'm not sure that it's working before. But now it's working, and we have just two minutes, please. So we need to.
Yeah, I'm almost done.
Yes.
So this is the way it works, yes. So in order to get better result, of course, this is not easy to realize. There are a number of challenges, and actually the way we improve the performance of the systems is by enriching the classification algorithms, introducing the brain network approach. And we can see that in a series of recent papers, we actually demonstrated that by integrating the way the different brain regions are interconnected, you can improve the classification performance and better understand how actually people learn to use this kind of new devices, which is not trivial at all for a naive person. So as you can see, actually we have these two lines of research, one theoretical on complex systems, the other more on brain-computer interfaces. In the end, we use one to enrich the other, and there is a very rich dialogue between these two research lines. We also have introduced new software that integrates and are publicly available, so people that wants to integrate and use network approaches for brain-computer interfaces can use this software. We are really pretty much involved in many translation and collaborations with the industrial partners in order to make our solution portables. And of course, we have a certain number of collaborations mainly with hospitals, because brain-computer interfaces, can have a strong impact on many diseases, especially the ones that are associated with handicaps like a stroke or spinal cord injury. We have a certain number of applications and projects that are funded by different national and international organisms. And with that, I end up here. Thank you very much.